Now, without further ado, let's get started introducing this morning's plenary speaker, Dr. Matthias Ullrich. Dr. Ullrich studied microbiology at the Friedrich Schiller University in Jena, Germany, conducted his PhD thesis on fire blight at the Max Planck Institute for Cell Biology in Heidelberg, Germany, followed by a postdoctoral fellow in plant pathology at Oklahoma State University in Stillwater, Oklahoma. Prior to 2002, he was an assistant professor for microbial ecophysiology at the Max Planck Institute for Terrestrial Microbiology, working on thermoregulation of bacterial gene expression. And since 2002, he has gone on to become a full professor of microbiology at Jacobs University Bremen, working in 4 major fields: marine microbiology, plant microbiology, antibiotics research, and plant metabolomics, including rhododendron and cocoa. Today, he will be telling us about a systematic approach to phytochemically evaluate the antimicrobial activities of a large subset of rhododendron species. And please take it away, Dr. Ullrich.
Okay, thank you so much, Juliana, for this very kind introduction. I'm going to share my screen and I'm going to start the slideshow. And I hope that everything is fine and you can see the slide so that I can go ahead. So thank you very much for the kind invitation, and I truly appreciate the chance that I can share some of our work with a number of rhododendron specialists here, and also looking forward to the feedback which I get, so please ask any question you might have. We were conducting a systematic approach to phytochemically evaluate the antimicrobial activities of a large subset of rhododendron species. And when I say we, I have to announce, first of all, also sort of as an acknowledgement, the team which was doing that actually. So we had a cell biologist, a phytochemist, a plant geneticist, and a botanist, along with a whole bunch of very talented and very enthusiastic PhD or master students, which are all shown down here, quite international, to work on a topic which was actually born in Bremen, here in Germany. And I will come to that in a second. But before mentioning that, you might see from the origin countries of the PhD students that Jacobs University is a truly international university. We have more than 110 nations here on campus. English is the main language spoken here, in contrast to many German universities. And we, of course, work on topics in an interdisciplinary fashion. So as you see, these 4 disciplines, or including myself as a microbiologist, 5 disciplines are quite diverse. So why rhododendron? Why did it become of interest to us? Prior to 2013, that's when we started this the rhododendron project, we had never been anyhow involved in rhododendron. However, in the Rhododendron Park in Bremen, we have the worldwide second largest collection, genetic collection of rhododendron species. And from the Rhododendron Park and its director, Dr. Hartwig Schepka, I learned that rhododendron, as you all know, is one of the species richest wooden plant genera on Earth. So when I learned about this, that we have a resource here, that we don't have to go to different places, Indonesia, Nepal, China, you name it, but we can draw the material from our botanical garden here. Plus, we find this high diversity in genetic diversity in one genus. We felt like it would be possible to take a larger subset of species, taxonomically defined species, and analyze them in terms of their antimicrobial potential, but also in terms of their phylogenetic relatedness, how close are they towards each other, as well as their cytotoxicity. Because whenever it comes to an antimicrobial agent which we want to use in medicine or in veterinary medicine or wherever, we always have to consider that rhododendron per se, from a chemical point of view, is a toxic plant. So I'll come to that in a second. So the problem which we were facing is that there's an increasing multiple resistance towards antibiotics among pathogens, all kinds of pathogens. There's a loss of effectiveness of chemotherapeutics. So that brings us more into the field of do plants like rhododendron have, for instance, preventive or cancerotherapy, chemotherapeutic potential. The approach which we chose for that was that, or the approaches which are possible to do is on one side chemical synthesis, rational design, or elaborative surveys and excursions to find novel bioactives. And that has, of course, been done numerously. And in this context, I would like to raise a point of concern, which many of you might have at the same time, since Nagoya has been signed by many countries of origin. We have, of course, also an issue here that even if we would conduct those excursions, that finding novel bioactives does not necessarily mean that we can make use of them without a lot of bureaucracy. So we can discuss this later on. The point is that the material which we have worked with from the botanical garden has all been planted in the botanical garden way before 1991. And that was, at least for Germany, and I don't know whether that is true for in general, that was sort of like the deadline beyond which material which comes from places of origin cannot be processed and cannot be valorized in an independent manner. So it was clear that we would make use of the biodiversity which we have on site, and that's why we collaborated with the Rhododendron Park. Rhododendron Park was happy about that because they consider themselves a place of pleasure, a place of education, but also a place of research. And that's why we are here. So what we also assume, and that was our hypothesis, that nature has been the best chemist ever. And if we find anything novel, if we find anything which has a potential as a chemotherapeutic or as an antibiotic, it would already be there, but we haven't found it yet. And if we find it, we have to make sure that we can isolate it in such a way that it does not contain harmful substances along with it, which is the major, how can I say, hindrance at the moment for any kind of plant therapeutics, that usually you have a mixture of compounds, and not all of those compounds have the desired properties, but many have undesired properties. So why particularly rhododendron? There are numerous ethnomedical reports and myths from China, from India, from Nepal, from Indonesia. Rhododendron leaves, rhododendron material has been used for thousands of years in ethnomedicine. And that's why it was reasonable to assume that we might find also something novel. There's a high biological diversity available in the Rhododendron Park Bremen. That was, of course, a plus point for us. And the genus is particularly species rich, which suggests a high genetic plasticity and diversity. In consequence, the ability for the scientists to differentiate between one species which has the antimicrobial substance versus another species which does not have the activity. So if we then would have those two closely related species which have different activities, and we would screen their genetic background for the reason for these differences, we would also learn more about the biosynthesis of the corresponding molecules. And that was, of course, from a scientific point of view, also of interest to us. So the phylogenetic origins of rhododendron are coming from the China, Nepal, India, northern India areas, mostly China. And the diversity which we can observe in moderate climate or alpine climate zone, mountainous climate zones, is of course highest, for instance, in China. We have at the same time also tropical rhododendron species, Vireya, as an example for tropical diversity. And here we have particularly high diversity in Indonesia. That is, of course, also from a or from a geobotanical point of view, very easily explainable with the island formation and with the separation of species at that time point. So both the tropical rhododendrons, we have a big tropical greenhouse in the rhododendron park, as well as the moderate and alpine species, we had a good choices to make from. So what was our approach? The approach was that we would take as diverse as possible rhododendron species, that we would conduct for all of them, and we have taken roughly 180 different species. We would conduct a phylogenetic analysis. We would test all plant extracts for antimicrobial activity. We would then, in the same moment, test those extracts for cell toxicity. And we would conduct with the corresponding extracts a high-resolution phytochemical analysis. The data would be integrated, and that was, of course, the prerequisite for everything, that we have the computing power to integrate, and also the informatics, the systems biology power to integrate all these this information and obtain from this bioactive species on one side. So which ones are more bioactive than others? If we dissect those bioactive species further, we would eventually find novel antibiotics. We would determine whether the extracts or enriched in antibiotics extracts would be safe in use or would be usable as anti-tumorigenic drugs. Because if a compound, for instance, is very cytotoxic, it always has the potential to act as an anti-tumorigenic substance. Finally, we would like to, shown here in the bottom right side, we would of course like to understand the chemical structure of those compounds. So the procedure for that is that we start with a crude leaf extract. It's a simple methanol extraction, and we would screen for antimicrobial activities. Based on that, we would find active extracts and disregard the inactive extracts. From that, we would do a fractionation, a chemical fractionation. We would use solid phase extraction, high pressure liquid chromatography, followed by mass spectrometry. And we have the abilities for this. Juliana was mentioning that we are working on rhododendron, on cocoa. For cocoa, we use this very similar approach. In contrast, we do not look for antimicrobial activities, but we look for characteristics for chocolate production, and we use the same procedure for that. So once we have done this fractionation, we will proceed, but before that, the active extracts have to be bulletproof in a sense that they are tested for cell toxicity, and we use for that two types of mammalian or human cell cultures. So we are working with cell cultures here. One of them is intestinal and one is keratinocyte cell culture. The intestinal one, because if we want to use the agent, for instance, as an oral drug, then the intestinal cell lines or cells need to withstand the corresponding extract. The keratinocytes are indicative for skin cells, and we want to, of course, also consider using some of these extracts as topical treatment of wounds, for instance, on the skin. So with high purity fractions, we would then do a fraction screening for antimicrobial activities. This is not really skyrocketing new work. This has been done in the past as well. The main difference for us is that the resolution we are achieving with the liquid chromatography mass spectrometry and the corresponding fractions allows, of course, a much finer resolution of fractions. Now, all these fractions, the active fractions, need to be screened again. And that is, of course, a lot of work. Again, we will get active fractions and we will test those active fractions for cell toxicity again. And if they are active but not cell toxic, or they are cell toxic but not active, then high purity active fractions can be obtained and they can be then used for different further analysis. The way of how this would end then at the is that we do a structure determination, and that currently is best done with LCMS, so liquid chromatography mass spectrometry, as well as nuclear magnetic resonance analysis, NMR analysis, which allows for, at least for smaller molecules like polyphenolic substances or monomers of polyphenolic substances, allows a relatively safe structure determination. I will give you an example where that has worked actually. So which rhododendron species act against microbes? That was, of course, our initial question. And for that, we had to, of course, set up the hypothesis that the phylogenetic relatedness tells us something about which species are more antimicrobial versus which ones are less antimicrobial. In this context, I would like to make a disclaimer, if I may say so. Since we are using the plant or the leaf material which comes from a botanical garden grown in northwestern Germany, we can only make statements about the antimicrobial activity of those species under these semi-artificial conditions. Now, rhododendron has been bred for centuries already in England. In northwestern Germany, we have a long tradition of growing and generating hybrids and even breeding new species. In consequence, many of those plants which we have actually analyzed have adapted to the conditions here. If they are still bioactive, then they would be, it would be good. But the disclaimer is on that if the original location where those plants are found in nature would provide completely different environmental conditions in terms of humidity, in terms of rainfall, temperatures, and so on, we can, of course, not deduce a general statement about the species. Yeah? But the phylogenetic analysis, the genetics, is, of course, safe, irrespective of whether we take the sample from China, from the mountains of China, or whether we take them here from the botanical garden. So the markers which we have applied were three major markers, and those of you who are familiar with plant taxonomy and rhododendron taxonomy will eventually know these corresponding genetic loci. This was one, we had another one, and then we had the third one. And those markers, of course, were known from literature to be the relevant ones in order to generate a phylogenetic tree of the plant material which we had here on site. The outcome of this was a beautiful and high-resolution phylogenetic tree. Can't really read that because it's too small print, but what it hopefully demonstrates to you is that we have subgenus structures, and these subgenus structures beautifully match to what's already known for rhododendron's taxonomy. With a set of these taxonomically identified or characterized species, we then conducted this antimicrobial assay analysis. What you can see here is a German written, sorry for that, a diagram which shows in red the killing of gram-negative bacteria versus in blue the killing of gram-positive bacteria. We used different gram-negative and gram-positive bacteria. We had a selection of them, pathogens and non-pathogens, plant pathogens, because the idea here was, again, that plant pathogens might be particularly adapted to toxins derived or antimicrobials derived from rhododendron. Now, rhododendron doesn't have a site of agrobacterium, and some Pseudomonas species doesn't have true bacterial pathogens. There are other fungal and insectal or nematodal organisms which cause insects and nematodes, which cause major problems to rhododendron, not so much plant pathogenic bacteria. Nevertheless, what we can conclude here is that some species, very few, show an activity against gram-negative bacteria. Now, gram-negative bacteria are characterized by a double membrane. They have the normal cell membrane, and then they have an outer cell membrane, which means that there are two layers of hydrophobic membranes which keep hydrophilic substances away. And what we learn from here in gram-positives, they have only one membrane, but a thicker cell wall, which is penetratable for hydrophilic substances, that the majority of killing, the majority of inhibition among the entire genus here is for gram-positive bacteria. We conclude from that we are talking about hydrophilic substances here. This is the summary of the bacterial species which we have taken. So a total of 26 bacterial species has been analyzed. And the idea here was not just to take two organisms, a gram-positive and a gram-negative, but take a larger number of diverse organisms. The background or the background question we had here, or the hypothesis we had here, is it possible to determine the the target range, not the host range, but the target range for some of these rhododendron extracts or bioactive compounds? Will the target range tell us something about the actual target inside the microbe? Because such an analysis of finding antibacterial substances is always associated with finding the structure-function relationship or the target. So this is the summary. And you can see that two representative pathogens have been included. We, in the meantime, have also analyzed a lot of more so-called level 2 or security 2 organisms. But Staphylococcus is a beautiful, not beautiful, but a very good gram-positive example, and Pseudomonas aeruginosa, of course, as well for gram-negatives. So when we take these 26 bacteria and we take the 180 or something like this extracts, we have to do a polyphasic approach. We use the principal component analysis. We add all the data for the level of activity versus the genetic background of the plant and of the bacteria. And with these PCAs, with these principal component analysis, we then can make some conclusions about what the majority of rhododendron species, which we have analyzed, acts against. And there we can see that there's a larger number of rhododendron species which act against gamma proteobacteria, and there are very few which also act against gamma proteobacteria. This gamma, beta, alpha, these are taxonomic affiliations of the corresponding bacteria. And we can see that there's quite a diversity. In other words, and that's what we concluded here, if we need a specific pathogen to be approached with a specific rhododendron extract, we would already have this information. The same can be done when we look at antimicrobial activity as a characteristic, as a feature, and we would talk about where do we find the activity. Then you would see that there is different types of the activity. So what can when I take one of those points here, just for your explanation, what this point indicates is that that's a subgenus Rhododendron species which has a characteristic pattern of inhibition of 26 different bacteria. Away from this, sorry, here's the cursor again. So away from that is this one here, which is also a subgenus, Rhododendron, but has a different pattern. Now those two here in the center, along with one Pentatera here, have a very similar pattern. So what we learn from that is that whatever substances are in these three species will be more similar to each other than to some of the other species. And what you please notice here is that the subgenus Rhododendron shows the most diverse pattern, whereas a number of other subgenera show a relatively similar pattern. It does not say anything about the intensity or the effectiveness, but shows us something about how close the rhododendron or subgenus, subgenera are to each other. So that is a very interesting analysis. What we can, of course, also learn about this when we then do the phytochemical analysis, which will come in a second. So what we could conclude from this is that we have a group of rhododendron species which show a very potent antimicrobial activity towards a very wide range of bacterial tester organisms. And the summary or the winners are, if you want, Rhododendron ferrugoneum, Ambigium, and Anthropogon. These three, which I have shown here as examples, were the top rhododendron species in our analysis, which showed the best activity. This is Ambigium, and this is Anthropogon. So in terms of their phylogenetic relatedness, as far as I know, they are all from the subgenus Rhododendron. So they all are already more related to each other than to many other rhododendron species. But they are, of course, still diverse in terms of their origin and in terms of their lifestyle. So once we knew which ones have the nice antimicrobial activity, we limited our research to a set of something like 50. And we then conducted an analysis in terms of which ones of these 50 active extracts are actually safe to use. Safe to use means, can we apply it to the skin of a human, or at least of a mammalian system? Can we eventually even swallow it and without getting toxified or poisoned? So the way of how our cell biologist did this with her students, master and PhD students, was an elaborative analysis of cell toxicity. What you see on this very colorful image here is a number of different cell cultures. So this is one line of cell cultures, this is one, this is another one, and this is a fourth one. And then we applied different concentrations. So that's a control line here, or column, sorry, column. 10 micrograms per milliliter, 50 micrograms per milliliter, and 100 micrograms per milliliter in some of those here. And then we observed how would these cells behave. Claudia Briggs is using a beautiful fluorescent dye and fluorescent antibody labeled system to find out which parts of a cell are affected, whether, for instance, the mitochondria or the energy conserving, not conserving, but converting elements of our human cells are affected, whether the nucleus is affected, whether the endoplasmatic reticulum, and so on and so on. All these cytoplasm, cell biological features are affected. As a summary for this, we could show now, talk about thousands of these beautiful microscopy images. The summary is shown on this slide here. And I have to guide you through a little bit what has happened. So we have restricted ourselves here to the 10 or so best performing rhododendron species. And then we have conducted for them 5 different assays, A, B, C, D, E. We have conducted these assays with 3 different concentrations. Down here you see 5 micrograms per milliliter, 50 and 500 micrograms per milliliter. 500 micrograms per milliliter means half a milligram per milliliter has been applied. And you would not be surprised that in this concentration, many of those extracts actually are cytotoxic. Black in this scheme here means toxic, so not suitable. Gray means non-toxic, suitable. And what we learn here is that when the concentration is lower, we have less toxicity. And what we also learn, and this is the differentiation between the upper part of the slide, where we have intestinal cells, versus the cytotoxic toxicity towards HACCT. These are keratinocyte cells. So skin cells withstand a higher concentration of most of the rhododendron extracts versus intestinal cells. For intestinal cells, you need to use a lower concentration of the compound. That's what we can learn here. If we are using 5 different assays, most of previous studies have used one of those assays, right? And then they have made with one of those assays, let's say take the assay A, they made the statement, it's safe to use. It's safe to use. And it's, in this case, even safe to use. at the highest concentration. So if you combine here, and I hope you can recognize what the way of how we approach this in general. If you combine the different assays and then you look at the picture, you will get a more comprehensive picture. But what you can learn from that is that those species here, including Phylogeneum, is suitable to use in 50 micrograms per milliliter on skin cells. Yeah? Why the toxicity is here in Phylogeneum with 5 micrograms per milliliter, we do not know. That has been the result, and it's hard to estimate what could be the reason for that. Yeah. Okay, so which of these substances, which of these extracts are containing which substances and which are responsible for antimicrobial activity? So this fine analysis, fractionation, which I outlined in the beginning. For this, we used high-resolution phytochemical separation. So now our phytochemist comes into place and generates a pattern of fragments which then can be tested for activity. This has been beautifully published in phytochemical analysis, where we show that there are cannabinoids from Rhododendron colletianum, which is another species, showed this clear bioactivity and antimicrobial activity. One short note, the reason why we did not focus on Rhododendron pherogoneum or ambiguum was that the first author of this study here, Inam, comes from Afghanistan, had a stipend from the German Academic Exchange Service, and was supposed to do something for Afghanistan. I don't have to tell you that Afghanistan is a very hot topic at the moment, a very sad topic. Inam used the material from the rhododendron park, which is known to be native to Afghanistan, in order to contribute to some extent to his home country. So what he showed in his work, which was of course a combined work of many other researchers as well, was that the activity, the minimal inhibitory concentrations in micrograms per milliliter are extremely low for some gram-positive bacteria when we take colletianum in comparison to some of the other also native to Afghanistan species which he could analyze. So that's why he focused on colletianum. He then conducted the very same analysis, which I have already shown previously, but not with 180 or so rhododendron species, but with 180 different fractions from colletianum. And then he finds, of course, using the PCA analysis, he finds the activity linked to a specific group of molecules, and then he purifies the molecules further. And the way of how this is done is a combination of chemical separation with a simple bioassay. And our simple bioassay was an agar diffusion assay. When you see an inhibition zone, that means the extract or the fraction is inhibiting microbial growth. If you don't see inhibition zone, that means that there is no antimicrobial activity. Based on that, Inam identified those structures here, which all showed antimicrobial activity and which all belong to the cannabinoids. So cannabis is familiar to everyone. You will be surprised that rhododendron contains it, but I've had a meeting, an international meeting, where a Finnish researcher also mentioned that in Finland, rhododendron is added, or extracts of rhododendron is added to beer in order to make that beer a little bit more funny. Yeah, so this to say. So despite the fact that, of course, cannabis has a biological origin, we find cannabinoid substances in all kinds of plants, including, as you can see here, in rhododendron. Now, interestingly, those compounds had already been described before and independently in a Japanese research work as being suitable to treat certain liver diseases. So in other words, they did not look at antimicrobial activity, but they found the same compounds. So that is sort of like an interesting anecdote. When we look at the corresponding concentrations of these cannabinoids, then we will find that gram-positive bacteria, including some plant pathogens like Clavibacter mechiganensis, are very effectively killed, whereas and even Staphylococcus is. So these are all gram-positive organisms, whereas E. coli is not inhibited at all. We need more than 1,000 microgram. So we're talking about, a very high concentration in order to treat E. coli. So just to put this in context, the European Union's threshold for a novel antibiotic is 32.5 microgram for a lead substance or 16 microgram for an individual compound. A lead substance would be a group of substances which can then be modified, chemically modified in order to make a stable antibiotic, for instance, out of this by the pharmaceutical industry. So you can see that we came across a good way. We found some. We wanted to continue with this, and we, of course, also published that work. This is published in Frontiers in Plant Sciences. We wanted to continue working on that. We applied for new funding. And then we got the comment, and that's also something for the discussion if you want. We got the comment, oh, it's a woody plant. It grows very slow. So why worry about it? Yeah, we cannot fund that. You know? The way of what prompted us then, and now I come to the end of my presentation, is that we started to miniaturize plant material by growing culli and also short shoot cultures. So we are doing that now in order to analyze, for instance, individual species in terms of their yield of the corresponding antimicrobial substance. We make use of the treatment of these shoots with, for instance, certain phytohormones in order to boost antimicrobial activity. That's what we are currently doing, but we are also lacking funding, so we can do that only on a very small flame. So as we say in Germany, we would like to have a big fire, but in this moment, we need to run it on small flame. With this, I'm pretty much at the end. I hope I am still in time and allow for some discussion. Thank you very much for your attention.
Thank you, Dr. Ullrich. That was really, really fun. I really enjoyed that. What a great presentation. Now, everybody, I see a couple of claps. Go ahead and give your internet virtual thanks. And if you have any questions, please put them in the chat right now. I have questions. That's always true. So I'm going to start while people are getting their questions. So first of all, it's really cool to think about the lepidotes being the group with the higher diversity. And people often say, you know, lepidotes, the lepidote scales are about anti-herbivory. And they're fragrant. So I'm wondering, do you know where these compounds are produced in the plant? And is there any idea that the lepidote scales might be a factor in that?
Yeah, it's an excellent idea, which we also initially analyzed. So we took, for instance, leaf material, we took fruit materials, we took scales when possible, and we analyzed the antimicrobial activity. So what I can recall now, because that also got published, Abhinandan, our phytochemical PhD student, published that with his supervisors. And I think he noticed that the leaf material contains comparatively little material in comparison to, for instance, sclerenchyma parts, and the scales would probably be sclerenchyma, if I'm not mistaken. But the simple amount, the biomass which we would get, and the way of how to separate it from it prompted us to focus on leaf material, which was easiest to work on. The idea from the botanical garden, the Rhododendron Park, was of course also to eventually valorize the entire procedure. And they wanted us not only to look at where is the highest concentration, but where is the easiest to extract concentration. So it is fair to say that normally those antimicrobial substances are more present in sclerenchyma tissue. It's accumulating there. And that is also going in line with the fact that often cytotoxic substances, which are produced by a similar pathway, for instance, they're part of a similar biosynthetic pathway, would also accumulate rather in dead tissue than in living tissue. Very good question.
Wow, that is fascinating. I'm so excited about this. We do have a question. Subgenus Hymenanthes, too, is very large in terms of number of species, but their antimicrobial compounds are not so diverse. Any thoughts about what could be the reason for that?
Yeah. Well, there's two possibilities. First of all, I don't know whether you've paid attention to the little rings which we put underneath those subgenera. For some subgenera, we had a large group of plants to investigate. For Hymenantera, let me go back there, because that might be, yeah, I mean, which one are we talking about? I have to look.
Hymenanthes.
Hymenanthes, right? So we had 56 out of 392 analyzed. And that's a relatively small number. Here we have 64 from 272, I think. So one simple reason might be that we have chosen the wrong ones. And it's always random in this particular case. We, as I indicated, we guided ourselves on the basis of how old are these plants, how long have they been in the botanical garden, which is a Nagoya issue, and we might have missed it there. The other possible scenario might be that other subgenera or species have been exposed to more pathogens than Hymenanthes. That Hymenanthes is somehow has evolved in an environment where there are less potential native or natural enemies. That would be one suggestion. This only talks about antimicrobial activity, not about cytotoxicity. And as I also tried to explain, we wanted to make a guided analysis. So we disregarded many species which had no antimicrobial activity and did not focus on them in terms of their cytotoxicity. However, the cytotoxicity would have been a fair alternative approach, that we start with cytotoxicity. Just much simpler to start with an antimicrobial assay. Yeah.
Very interesting. Okay, we have another question from Connor Ryan. Do you think breeding would be worthwhile to improve yield? For example, cross Phytochemically with a fast-growing lepidote and extract antimicrobial compounds from the hybrid. Would you expect the antimicrobial to show up in the hybrid?
Yes, very, very, very good question. And I think if Hartwig Schepka would be here, he would immediately laugh out loud because he exactly proposed this, and he interacted with, or he works together with a plant breeder here in Northwest Germany, one of the leading ones, Timo Schröder. I don't know whether anyone has ever heard of him. He has a huge rhododendron farm, and they started doing this hybrid breeding. And they generated a hybrid, which was then planted many times in the greenhouse. I went there, took them from the greenhouse, brought them here into our very improvised plant facility, maintained them, this hybrid, for about 1.5 years, and then suddenly, and for whatever reason I could not understand, the plants died in our facility. You know? The plant breeder, noticed that the hybrid is not really selling well in his hands, so they gave up on it. However, this is exactly the point where we want to do the in vitro cultivation. We've started a collaboration with a group in Hannover. and Traut Winkelmann is a name. She has very good experience with these in vitro cultures, and we did a number of series of experiments already in order to test for antimicrobial activity. So we did not obtain enough plant material in order to conduct a thorough analysis. Our initial results of the hybrid was that they had the same activity as the previous one. We also did not notice a much better growth, and for sure not a more robust growth. And a more robust growth would have been one which withstands even the harsh conditions of our indoor. We have an indoor facility where we can manipulate the light and the temperature to some extent, but outdoor is of course much better.
Plants always like it better outdoors. It doesn't matter
Yes.
how nice you make your little spot for them inside.
Absolutely.
You said something interesting about people mentioning that it's a woody plant, so why bother? And actually, I hear that a lot doing evolutionary studies. So I push back against that because I thought, well, you know, because these are the ones with the interesting features, you know, not Arabidopsis becomes boring at some point. Your micro propagation technique is very interesting to me. So can you get, say, hundreds of individuals all in the same sort of stage and size using that technique?
We cannot because we don't have the facility, but I know a company which is called Pico Plant, P-I-C-C-O Plant, Pico, or no, so P-I-C-O, Pico. Pico stands for very small, right?
Yeah.
Plant. And they are located in Oldenburg, and they produce these shoot cultures permanently so that they have sort of like they want to have a somatic multiplication of not only rhododendron material. We included them in our proposal. Yeah. Irrespective, the proposal got rejected by people who were convinced that if you work with a tree, it will never pay off.
Yeah.
It's amazing.
We need to fight again. Keep fighting the good fight. I'm there with you because
absolutely.
Yeah. Here's another great question. Is there any relation between temperature altitude and the diversity of antimicrobial activity? You kind of alluded to this, that you're looking at garden plants. So do you have any idea about how different environmental factors might influence the compounds?
Yes, I'm pretty sure that these environmental factors will have a profound effect on the antimicrobial potential of plants. Not surprisingly, in Nepal, for instance, they are using teas made from rhododendron. They are using paste and extracts, different boiled extracts, in order to treat infectious diseases. And they can do that. So in other words, I cannot tell from our analysis because we have not compared them, but we have noticed that some species which are described to be active in nature did not show up in our analysis, which demonstrates the sort of like artificial or in vitro approach we have chosen. Nevertheless, we believe that if we learn about the biochemical synthesis of those compounds, that we can then go back to nature and screen for some markers which then tell us, okay, at this position, the material would be more suitable. By the way, was also a negative comment. I mean, you will not be able to deforestate the Himalaya for your purpose. And they are right? I mean, we just have to remind ourselves of Taxus, the Taxol, this very potent anti-cancer drug, which is nowadays produced, synthesized in vitro, of course, but where they had also calculated that you would have to deforestate the entire state of British Columbia and also Washington in order to to treat a couple of patients, efficiently. Yeah, that's. I mean, these are arguments which they bring about when it comes to such beautiful species like Rhododendron, this beautiful genus, right?
And that brings up, you talked a little bit about the Nagoya Protocol. And I think as a human, I fully agree with this. And I think it's a great thing to mind how we use resources that belong to others. But it is presenting a challenge for us as scientists and finding a way forward. So when you think about those plants that came between 91 and now, what is the fate of those plants? Do you have any thoughts about that?
Very, very good question. I mean, the Nagoya Protocol does not only affect our research in Rhododendron, but also affected the research in our marine field. I think it's it's not convenience. I think it's nerve breaking. It's avoidance of nerve breaking activities. Mm-hmm. I know from our marine research or collaborative marine research that where we tried to sample in Indonesia, right, in modern Indonesia, not before 91 or so, and that was a nightmare to just get to the right person who is responsible in Indonesia. Yeah. So this person has, in the course of the preparation of the project, the person had changed 3 times, and each time there was a different background of what you had to consider, and right. So on. So I think many of you might have made similar experiences. Don't want to be sounding too frustrated, but I think these materials are lost for us. Yeah. At the moment, unless the countries of origin would come up and say, please collaborate with us. Yeah. Yeah. And some do. But then, of course, the bureaucratic burden should be on their side because it is almost impossible for us here from Central Europe to communicate with people, for instance, in China or in India easily on this bureaucratic level, right?
We need discussion. We need people talking to each other. We need people working together. I think that's sort of what you're hinting at, is that we all need to put a concerted effort into, I think, recognizing that the materials are important, especially when you think about some plants in collections won't be in the wild very soon. And so these are important plants a lot of the time, and it would be a shame for them to be lost permanently. Absolutely. So it's just going to take some time for us to get that work going.
Yeah. Yeah.
Wonderful.
Well, I mean, if we had a strong lobby, right, or stronger lobby, that would already be helpful. Yeah.
Well, I don't know how you and we're going to get the world to cooperate. How about that?
Okay, that sounds really good. Yes. I mean, we try to convey the message also with our students, right? I mean, as you could tell, except the Egyptian student, everyone came from a country, if I'm not mistaken, where their rhododendron is grown natively, right? And
yeah, even in collaboration. Yeah.
Yes, and even in northern Africa, I think long time ago there were rhododendron forests, right? Yeah.
Really? I need to find out about that. Mm-hmm. I'm a history buff as well as a scientist, so I'm going to have to go check that out. Thank you. I would like to give you thank you again, a round of applause before we move on to the next section. That was a great talk. Really enjoyed it. Great Q&A too. Thanks, everybody.
Thank you very much.